talk about if you start with a basic biological neuron. And neurons have all sorts of shapes and sizes. If you have something that might seem typical to a pyramidal cell, you might see this particular structure with the core soma, a single axon, larger dendritic tree, multiple inputs coming from other axons. You might ask, well, how do I begin to start thinking about what's going on in the biological terms? Well, fundamentally, you're looking at this is a cell, okay? And so it's like lots of other cells. There's going to be an inside and an outside. But the interesting part about neural structures is you're going to be spending a lot of time looking at the various ions. And in this case, we're talking about charged ions in water. You're usually talking about a whole range of concentrations of how much of these ions would I get. And the typical ones you would get in a biological neuron, and these are chemicals you might be familiar with, is sodium, potassium, calcium, and chloride. In fact, if you're thinking about it, if I were to usually put things into this, it's usually things like, say, sodium chloride, which you might know is salt, or potassium chloride, which is a different kind of salt. But these are ones that we're used to seeing, and certainly calcium chloride is a form of a salt as well. And these are all ionic bonded elements. And so that means that when they get into water, the ions, because they basically pulled things into their, pulled things or gotten rid of the extra electrons, what will happen is they're basically acting like free carriers. They're kind of like hanging around things that keep them electronically neutral. But it's basically kind of a sea of ions and a sea of other ions, and everything kind of fits together that way. Which is kind of interesting if you come from an electrical engineering background, because now it looks like I just got a flow of charge. And it feels like, well, I've done flow of charge before, right? I mean, I can see this to say in semiconductors, I'll get electrons and holes. Well, this is not a shocking thing to think about of what's all happening here. So I've got a whole set of ions everywhere, and then I've got cells where I've got an inside and I've got an outside, and they're separated by a cell membrane. And it's really interesting because a lot has been understood about a cell membrane. In fact, it's cool that the very first cell membranes were actually constructed artificially in the early 1970s. That was work done at UPenn, very, very interesting work. And it gives us a good sense of what's going on. And these membranes are called bilipid membranes. And they're basically bilipid membranes because I have basically two sort of lipid molecules where one side doesn't like water, or one side likes water, and the other part of it doesn't like water. And you're like, oh, this is an interesting problem. Except that the two parts that don't like water hang out together, and the parts that like water sit near the water sides. And they can kind of go in this sort of nice little structure where they all pair up. And so all the ones who don't like water are all good. And all the ones who like water are all still happy as well. And so you get this nice sort of very tight bonded structure. At least it looks very tight. Now people may wonder what's something you might have seen of a bilipid membrane. And the easiest example is if I blew bubbles. And some of you may remember this from being a child or from an adult. It's always fun to do. That's actually a form of a bilipid membrane, and you might go, well, that's interesting. I blow it and it eventually breaks. Well, at the sizes of several centimeters, yeah, they might break, but at the sizes of, very, very tight, small, thin things in micron dimensions, quite strong, works very well. And so that's where you begin to look at this. What's interesting when you look at this is that I'm going to have charge on both sides, and so that's great. I can think about electrically as having charge on two sides of a capacitor. And in fact, it exactly acts like this because it looks like I've got an insulator and two elements there. So much so of an insulator that actually I also don't get any current through this. I mean, so you're usually talking about less than a picoamp of current for a several microns of size device. And so really a good insulator to work with here. And so you go, that's great, except that if it's a great insulator, why would I use it for any sort of calculations, computations? And what happens is there's other piece that'll show up, which is talking about what happens if I have a channel. And if I have a channel in the membrane, that actually allows ions to flow on both sides. And now what I've effectively done, in addition to having capacitance, is I may have created a voltage through this because there will be some sort of voltage control barrier. For those who've actually done semiconductors and think about p-n junctions or transistors, you're getting a similar factor in there, but there's also a conductance. And so now I have to think about that too. Or I get a chance to use that, and that conduction may be a function of the membrane voltage, the inside voltage that will change that conductance. And that's one way to think about this sort of very simple structure. It's a lot more complicated than that in terms of what's all going on, and is this really even a conductance, or how should I think about this? But what you begin to realize is that this kind of sets the stage of all the things that you could imagine. And the sort of simple picture, which goes back many, many decades, gives us a really good framework to sort of think about the sort of biological systems, but they're actually really interesting from an engineering and electronics perspective. And so all of these worlds come together in some really, really beautiful and nice approaches.